So this is Bio 211. Welcome to Anatomy and Physiology 1. We are looking at the nervous system. And up till now, what we've done is, I'm going to use this cartoon here. We have built multipolar neurons to understand how it is we can actually have specific types of potentials found on the dendrites in the cell bodies, unique types of potentials that travel, in this case, alongside myelinated neurons to the axon terminal. And what you're looking at below this, of course, is how much ligand or neurotransmitter is required to get to minus 55 for an action potential to occur at the axon and axon terminals, right? So, and this cartoon, summation is working under the assumption that you understand that at any given point in time, a neurotransmitter could be excitatory, which is just what, it's an NT. Excitatory, which is what the plus means, and, I should say or, not and. Or it could be inhibitory. We'll get to when neurotransmitters can be excitatory and inhibitory. Okay? And that's assuming what you're looking at here is that 1 and 2 release neurotransmitter quanta here, and the sum of those together gets us to minus 55, and then we start what's going to happen down here on this axon of number 3. Okay? Get it? Okay, well, we want to then start thinking about what are neurotransmitters. Okay? And to do that, we need to first erase this. We think about this. Both excitatory and inhibitory neurotransmitters are present inside the central nervous system and peripheral nervous systems. And this is the tricky part. Some neurotransmitters can be excitatory in some locations and inhibitory in other locations. What I want to make sure you do is first understand that neurotransmitters bind to receptors. And then we'll worry about what they're doing exactly where. You'll see this is really much more important inside chapter 15 and 14 than it is anywhere else inside the body. Okay, so what are the neurotransmitters I want you focusing on? Think about what's inside your textbook. I definitely want you learning what acetylcholine is. I definitely want you learning what glutamate is. I want you to know what glycine is. And all of these guys down here, collectively, these are biogenic amines. And you're going to see epinephrine and norepinephrine so much that there's just no way to not understand what it's doing. And dopamine, although it's really cool to think about, we don't spend a lot of time thinking about it inside AMP. You want to learn more about dopamine, even though it's a biogenic amine, you'll have to take a neuroscience course for that. Okay, so I've told you that there are a subset. I got 1, 2, 3, 4, 5 neurotransmitters you're really thinking about. Okay, and what do they look like? Well, Mother Nature did us a favor, as usual. She makes them from large to small, or small to large. And that's the way I want you to think about them. Small molecule neurotransmitters, acetylcholine, amino acids, glutamate, aspartate, gamma-aminobutyric butyric acid, and glycine. These are all small. The biogenic amines, they're a little bit bigger, right? And these biogenic amines are norepinephrine, epinephrine, dopamine, and serotonin. Notice I didn't include dopamine and serotonin in LISP before to study. ATP is a neurotransmitter, and that means that purines are neurotransmitters. That's the way to think about it. ATP has ribose sugar, adenine group, and 3 phosphates attached to it. Okay, this sounds familiar to you. You should remember that this is adenosine triphosphate. The example that I give for neuropeptides inside class, if I haven't already, is this is substance P. Look, it's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 amino acids long. Okay? And the only thing that's significant about this, aside from the example that I give inside class, is that it's big. It's really big compared to these guys. The other one that I like people thinking about is endorphins and enkephalins. These are the body's own natural pain blockers. So if you have substance P, which is a pain potentiator, pain is good. You need to know you're in pain. It's what keeps you alive, right? You have pain blockers, enkephalins, endorphins, and these allow you to now respond and keep your body functioning when you're experiencing pain. But how do they work? Okay? Neurotransmitters will either be agonists or antagonists. And agonists are anything that will enhance the neurotransmitter effect, or an antagonist, anything that blocks it. well beyond the purview of this class or the responsibility of this class. Is neurotransmitters will be acting antagonistically with one another. And this is really important inside chapter 15, but neurotransmitters can be modified. Their synthesis can be stimulated or inhibited. The release can be blocked or enhanced. You can see that Mother Nature can nuance this really simple idea of turning something on or turning something off. Okay? What I want you to pay attention to are this really simple idea that acetylcholine is released by many peripheral nervous system neurons and some central nervous system neurons. And it's excitatory on a neuromuscular junction, okay? Meaning if we're using our lower motor neuron to activate skeletal muscle tissue, okay, it's excitatory, but it's inhibitory at other locations. So this is one really important example of a plus and a minus. Okay, I mean, it's excitatory at one place. Just make sure you get that. Oops, let's go back. Control D, there we go. Let's go like this. All right, and it's inactivated by acetylcholinesterase. Let's go like that. A-C-H-E. Okay. Amino acids. I know you can think about there are many amino acids inside of the body. Glutamate is released by nearly all excitatory neurons inside the brain. Okay, and it's inactivated by glutamate-specific transporters. So this is something that we're not even going to entertain. Okay, but you have to shut off the transporter to stop the actual production of this. The example for gamma-aminobutyric acid, of course, is an inhibitory neurotransmitter for 1/3 of all brain synapses. So this is why the drug Valium is so effective at enhancing its inhibitory effect of GABA. Okay, now that might be fancy words for saying, hey, look, here are some examples of small molecule neurotransmitters, but what I want to make sure you understand is that Mother Nature has a mechanism for regulating all of them. So how do we actually use this? I'm going to pop down here to this one cartoon where Mother Nature creates circuits. Okay? And as long as there's a signal that starts the circuit, it continues. So this is a diverging circuit. Okay? So you have 1 goes to 2, 2 goes to 4, and this is a positive feedback system. Likewise, converging circuits. All these traveling into one output, converging to one point, reverberating. And this is, think of this as when you can't let go of a thought and it just keeps coming back. Think of like a double mint gum or something like that. Okay. It doesn't really matter which one you're thinking about here. Just notice that ultimately Mother Nature's figured out a way to regulate all of this. And in doing so, we can start building something like, hey, this should make sense now. Ball on stick, and let's change color to blue. Where'd you go? Ball and stick. Ball, stick, change blue. Travel in. Yay. Travel up, travel over. Let's change to purple. Purple. We have interneuron 1 at the thalamus, interneuron 2 at the cortex. Now red travels down, where our upper motor neuron travels down, decusses over, activates inside the spinal column here, yay, and then the lower motor neuron exits. Okay, so you can see how by building something, okay, you can actually get it to work. Now what you want to do is, of course, go to this last slide. Before we get there, I want to actually entertain some idea. Inside your textbook, let's get rid of the cartoon first. Your textbook, they say that you can actually have plasticities maintained throughout life. Yeah, you can actually make new dendrites now. There's no doubt about that. We can actually see that you can actually make new neurons as well. So when I'm teaching you something new and you do something like exercise afterwards, or more importantly, get a really good night's sleep, right? You can actually maintain not only the dendrites and axon terminals, but also the neurons that have been grown as well. Okay, it says limited ability of regeneration. In fact, these days I would go so far as to completely remove that from your textbook. Okay, there are definitely some instances where that can't happen. Traumatic tissue injury to large parts of your central nervous system and peripheral nervous system are non-repairable. You can't come back from that. But for the most part, what we're learning, specifically when it comes to learning, is you can literally change the complexity of your brain by increasing the difficulty of the work that you're attempting to do. So hopefully that helps you understand why some of the questions are a little bit more open-ended, such as this one now. It's the end of chapter 12. Now, what do you need to do to make sure you understand this? And if I'm you, I'm thinking to myself all the way back to the very beginning of chapter 12 and thinking, okay, well, I know that what makes this cell type distinct is it's excitable. And that means I need to understand excitability. And how do I regulate excitability? And, let's go back to like grade school. By the time you finish this chapter, you're not just thinking about one neuron, you're thinking about multiple neurons, right? So one neuron, next neuron, next neuron, next neuron. How do they actually communicate with one another? And then what you can do is you can start thinking to yourself, well, I need these three to become excited, but I need this one to be inhibited. So you can work through now the dynamics of going from a graded potential to an action potential. Notice how I'm changing the cartoon just a little bit. Okay, I'm multiplying that all the way through, but if I'm going to inhibit this one here, I'm going to have to make sure my graded potential now looks like this, and that there is no action potential. Okay, that's the way you want to start thinking about it. Okay, cool. Here's to making new neurons. Have a great day, guys. Bye.